Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about rolling circle replication. The rolling circle replication was first described nearly half a century ago while analyzing the replication of E. coli phage 5X174. Since then, many other genetic elements replicating by rolling circle mechanism have been identified, including bacteriophages, plasmids of gram-positive and gram-negative bacteria, archaeal plasmids, and eukaryotic viruses. Rolling circle replication is an asymmetric process. The replication of leading strand and lagging strand are initiated from different replication origin. RCR initiator proteins play a key role in the replication of leading strand. The RCR proteins cleave the leading strand within the replication origin for initiating the process of replication. RCR proteins are divided into three groups. They are Rep protein, MOP protein, and TNP protein. Rep protein is responsible for vegetative replication. MOP protein is responsible for mobilization for conjugal transfer, and TNP protein is responsible for transposition. The best known RCR replicants are bacteriophages and bacterial plasmids, including 5X174 and another plasmid PT181 that has been isolated from Staphylococcus aureus. The rolling circle replication is an asymmetric process. Two different replication origins are required for the initiation of replication of leading strand and lagging strand. These two different replication origins are DSO and SSO. DSO means double-stranded origin and SSO means single-stranded origin. Double-stranded origin is required for the initiation of replication of leading strand, whereas SSO is meant for initiation of synthesis of lagging strand. In both 5X174 and the plasmid PT181, the double-stranded origin contain two different sites. These two different sites are nick site and the bind site. Replication initiation protein interact with both nick site and bind site. First of all, replication initiator protein interact non-covalently with the bind site. After this, the exposed nick site is cleaved by the initiator protein, thereby releasing the 3' end of the DNA. This 3' end will be used as a primer for the synthesis of leading strand, and the replication initiator protein remain covalently attached with the 5' end of the nicked DNA until the replication of the leading strand is terminated and the circular single-stranded intermediate is released. Friends, this model is based on the study of rolling circle replication in the plasmid PT181. During the rolling circle replication of this plasmid, two different replication origins are required. One is double-stranded origin. In this region, double-stranded origin is present. This double-stranded origin contains two different sites. One is nick site and the other is bind site. Whereas another replication origin is present, that is known as SSO. SSO is meant for replication of lagging strand. In this diagram, the black arrows indicate the direction of replication, whereas dashed red line indicate the nascent DNA or the newly synthesized DNA. And here you see these two are the replication initiator protein that has been covalently attached with the 5' end of this nicked DNA. And this is the 3' end. This 3' end is used as a primer for the synthesis of new DNA strand. After this dotted black arrow indicates the nucleophilic attack of the catalytic tyrosine residue on the phosphodiester bond of DNA, or it can also indicate the nucleophilic attack of the 3' end on the phosphotyrosine bond between the enzyme and DNA. Before understanding the mechanism of rolling circle replication, let us know about rep proteins. Rep proteins are replication initiator proteins that process specific single-stranded DNA sequences to initiate rolling circle replication and hairpin replication in viruses. Rep proteins are the member of HUH endonuclease superfamily. HUH endonucleases are a group of endonucleases that can process single-stranded DNA sequence. The processing mechanism involves cleavage and joining of DNA fragments in order to facilitate various biological processes, for example, rolling circle replication, rolling hairpin replication, bacterial conjugation, DNA transposition, and DNA integration into the host genome. The name HUH endonuclease has been given because all of these enzymes possess a conserved histidine hydrophobic residue histidine motif. So friends, we are discussing about the mechanism of rolling circle replication in the plasmid PT181. So PT181 is a single circular double-stranded DNA molecule. It possesses two different replication origin. This is the double-stranded origin, and here this is the single-stranded origin. Double-stranded origin is useful for initiation of replication of the leading strand. And here the replication initiator protein is Rep-C protein. Rep-C protein is a type of replication initiator protein. It exists as a dimer. Each monomer of the Rep-C dimer possesses a catalytic tyrosine residue. So a Rep-C dimer contains two catalytic tyrosine residue. One tyrosine residue known as Y1 and the other is Y2. Y1 initiates the replication, whereas Y2 terminates the replication. So this is about Rep-C dimer, and this is the double-stranded origin of PT181. First of all, the nick site which is present in double-stranded origin will be cleaved by the Rep-C dimer with the help of catalytic tyrosine residue. So the 3' end will be free. This is the 3' end that will be free. The freed 3' end will act as a primer for the synthesis of new DNA strand. This red colored strand is the new DNA strand that is being synthesized. During the process of synthesis of the leading strand, the 5' end of the nicked DNA remain covalently attached with the Rep-C dimer. This is the Rep-C dimer which has been attached covalently with the 5' end of the nicked DNA strand. And during this process, the nicked strand is displaced. This is the new DNA strand that is being synthesized by using the 3' end as the primer. At the same time, the 5' end of the nicked DNA remain covalently attached with the rhapsidimer until the synthesis of leading strand is completed and the single circular DNA intermediate is released. After this, there are several trans-esterification reactions. This trans-esterification reaction is initiated by Y2. The dotted arrows indicate the nucleophilic attack. Rhapsidimer facilitates the process of rolling circle replication by processing specific single-stranded DNA sequence. During processing, depending on the situation, rhapsidimer make and break phosphotyrosine bond and phosphodiester bond. Phosphotyrosine bond is present between the catalytic tyrosine residue and DNA, whereas phosphodiester bond is present between adjacent nucleotides in a DNA strand. So friends, this is the 5' end of the displaced DNA strand. This DNA strand is the displaced DNA strand. The 5' end of the displaced DNA strand has been covalently attached with the rhapsidimer. This bond is the phosphotyrosine bond. And another bond that is present between nucleotides in a DNA is known as phosphodiester bond. So during the course of synthesis of the leading strand, there are several transesterification reactions. The first transesterification reaction is initiated by Y2. In this transesterification reaction, the catalytic tyrosine residue Y2 it make a nucleophilic attack on the phosphodiester bond. So the 3' end become free and another phosphotyrosine bond is formed between the newly synthesized DNA strand and the catalytic tyrosine residue Y2. This is the first transesterification reaction that is initiated by Y2. After this, another transesterification reaction initiated by free 3-hydroxyl group of the DNA. This is the displaced DNA strand and this is the 3' hydroxyl group. This 3' hydroxyl group will make a nucleophilic attack on the phosphotyrosine bond. So the phosphotyrosine bond will be broken and the displaced DNA molecule will be freed and it will be circularized to form a single-stranded DNA intermediate. Further, there are some additional reactions that occur during the synthesis of the leading strand. So the DNA synthesis is terminated and also DNA processing occur with the help of Y1 catalytic tyrosine residue. And finally, there is another transesterification reaction. In this transesterification reaction, the nucleophilic attack is initiated by the free 3' end on the phosphotyrosine bond that is present between Y2 catalytic tyrosine residue and this DNA. So this phosphotyrosine bond is broken and this rhapsidimer become free and the two ends are joined to form a circular DNA strand. So a new molecule of PT181 is formed. The conversion of circular single-stranded DNA intermediate into double-stranded form is initiated at the replication origin of the lagging strand that is known as SSI in 5X174 and as SSO in the plasmid PT181. SSI means single strand initiation site, whereas SSO means single stranded origin. Rhapsi protein are not involved in the process of synthesis of lagging strand. So synthesis of lagging strand takes place with the help of enzymatic machinery of the host. A primase complex is recruited within SSI. The function of primase is to synthesize RNA primer. Starting from SSI, the primase move along the DNA and periodically synthesize RNA primer. Whereas in case of the plasmid PT181, a single primer is sufficient to continue the synthesis of the lagging strand. So, with the help of enzymatic machinery of the host, the circular single-stranded DNA is finally converted into a double-stranded DNA. So, one more molecule of PT181 is generated. The rolling circle replication of 5x174 and that of the plasmid PT181 differ significantly in the leading strand termination stage. In 5x174, the termination of replication of the leading strand initiate another round of replication in order to enable the virus to rapidly amplify its genome before lysis of the host cell. But in case of plasmid PT181, because its copy number is strictly controlled, so after completion of one round of replication, further replication of this plasmid is not permitted. This is due to inactivation of the Reps protein. Friends, this is about rolling circle replication. It is a somehow complicated process. So you have to watch this video at least twice or thrice so that you can understand the process. Friends, molecular biology is a difficult subject but is very interesting. By studying molecular biology, you can understand about life at the molecular level. Friends, please share and like this video and also subscribe my YouTube channel known as Sam Online Class. Celebrate Azadi Ka Amrit Mahotsav. Thank you very much. See you again. Bye bye.